Thank you, Natasha, for organizing this exciting, session and inviting me to present our work. Today, I would like to, describe a recent work in the lab, engineering remotely controllable CAR T cells for cancer immunotherapy. So as we know, CAR T cell become very popular in treating cancer. In general, the method is you taking the blood from patient and genetically modify them to express the so-called chimeric antigen receptor on the surface of these T cells. And this chimeric antigen receptor now have these single-chain antibody, on the surface, which can recognize antigen on the tumor and therefore attack tumor cells. So it has been revolutionary and very successful for blood cancers because there's a very clean, CD19 antigen to be target. However, for solid tumor, it still has some, issues, particularly on target of tumor toxicity. There are different ways for people to engineer, particularly utilizing synthetic biology to increase the precision of these CAR T cells so that they wouldn't cause, damage to normal tissue organ. However, because the physiology and our body is very complicated, there's almost no possibility to engineer perfect antigen antibody pair. So some of the CAR T, when they are fused back in the patient, they will go attack the tumor, but they also attack like normal tissue organ, even those tissue organ express minor level of the antigen. So we're trying to use a new technology, so-called focused ultrasound controllable CAR T. Essentially, our understanding would be to engineer these CAR T cells so that they can be only activated by focused ultrasound at a particular location around tumor. So the CAR T can go everywhere, but they will have no damage to the normal tissue organ because only those at the tumor site exposed to ultrasound will be activated. So we utilize this concept mainly because ultrasound essentially is a mechanical wave that can penetrate deep in tens of centimeters in the soft tissue. And if you can focus the ultrasound beam, you can create, strong energy to cause the glass breakage. And we try to utilize this heat shock promoter to drive the gene reporter as well as CAR T, so that ultrasound focused to these cells will generate temperature increase, for example, to 43 degrees, and then they can turn on this heat shock factor to be recruited to the heat shock promoter to drive the gene production. For this case, then the reporter can be turned on. And we first demonstrate this concept indeed works. As you can see on the control, these ultrasound controllable T cells, no expression of the gene. Now with ultrasound to raise the temperature to 43 degrees, this GFP reporter can be turned on. Even better, because ultrasound can be precisely controlled with timing and location, we can create this so-called short paused ultrasound pattern. Only 5 minutes ultrasound and temperature increase, and for 3 times, it's like short pause pattern or ultrasound, we can induce the gene expression like more than 90%. Suggesting it's a really efficient way to control genes. And this is particularly good because this short paused ultrasound wouldn't cause any toxicity to the T cells, because we know human, T cell, primary T cell particularly, they are quite delicate and sensitive to the heat. So if it's a long-term heat, they may cause damage to the T cell themselves. We further demonstrate these short paused ultrasound can indeed be applied to control the gene expression. So we engineer this inducible gene driving the CAR labeled by GFP to show the location. So basically this genetic cassette, we have constitutive promoter expressing M-cherry, which label the engineered T cell. And we have this inducible CAR cassette fused to GFP. And results show that indeed, without ultrasound stimulation or temperature raise, this engineered cell representing by the M-cherry signals, they don't have any CAR expression. Now, if we apply the stimulation, and these engineered cells now start to produce CAR on the surface, as you see this ring structure on the surface of these cells. We then apply this concept for in vivo studies. In this case, we integrated focused ultrasound together with MRI imaging machine. So the focused ultrasound-induced temperature can be monitored by MRI imaging, and then it will provide a feedback to control the power of this focused ultrasound so that the temperature and pattern can be precisely controlled. These are images of ultrasound transducer on the top view. As you can see here, these are transducer, array on the surface. And we can also look at from side view. You can see these ultrasound beams can be steered in the X, Y direction as well as in the Z direction. Because ultrasound can penetrate tens of centimeters, we can steer them to cover anywhere in the body of the mice. And these are the images of the MRI to show the temperature. So this temperature will provide feedback to the control of the ultrasound so that this temperature can be precisely controlled at a different location. And the results show this system indeed work really well if we introduce ultrasound controllable cells in the mice without ultrasound stimulation, nothing. After we ultrasound, shine to these cells, even with like twice or 5 minutes short pause stimulation, you can see the gene production very well. Now we further tested whether this ultrasound controllable CAR T works in vivo. We introduced in the same mice 2 tumors on the left and right sides, and then we introduced ultrasound controllable CAR T on both sides. And then we only activate the left side with ultrasound, leaving the right side as a control. And results show that with one dose meaning 5 minutes for 3 times, we can already see a significant suppression of the tumor on this left side. On the right side, that's the control. The difference is obvious. And these are images, as you can see on the left, these are ultrasound activatable CAR T. On the right, this is the control, exactly same mice, left and right. Okay, and after 18 days, we can really see very strong, suppression of the ultrasound effect. So these are on the lymphoma tumor cells. We further tested in prostate cancer. We again do the same thing. We introduce prostate PC3 tumor cells on both sides. We introduce then the PSMA expressing CAR on the both sides, and then only activate the left side. Then the result was also quite clear, similar to the lymphoma tumor cells. Only one dose ultrasound stimulation, we can already see a drastic suppression comparing to the control. Again, this is the left side of the ultrasound treatment, right side is the control, exactly the same mice, left and right. After 18 days, we can see significant difference. We finally tested whether all focused ultrasound controllable CAR T FAST-CAR is more or less toxic against the off-tumor targets. So we did this experiment. We introduced cells expressing antigen on both sides, and then we introduced locally the standard CAR T on the left side and monitor the growth of the target cells. As you can see, even we locally introduce the standard CAR T on the left side, it attacks both. So this is the time course on C, distal tumor cell growth, proximal cell growth. You can see after like 20 days, the standard CAR T attack both of them, kill both of them. And for the FAST-CAR, we do the same thing. We have antigen expressing cells first injected, and then we only locally inject the FAST-CAR controlled by ultrasound, and then only activate on this left side. And you can see this distal side on the D, distal side, no effect, and the proximal side precisely killed, suggesting all technology can indeed allow us to have higher precision, only kill the specific tumors. So that's our dream. We hope we can engineer these different, ultrasound controllable CAR T cells so that we can have a jacket, for example, with transducers, and these transducers can be controlled by cell phone and program so that we can precisely control the timing. Like, for example, a patient can be massaged, let's say every week for only, once or twice, and then they can precisely control and kill the tumor in the particular locations. I have to thank my lab members, particularly Dr. Ziliang Wang and Molly Allen, who engineered different genetic set, and also Dr. Yijia Pan and Shirley Wu, who worked out these ultrasound controllable CAR T cells. I also want to thank our collaborators and funding support, mainly from NIH and the Department of Bioengineering at the UC San Diego. I would be happy to answer questions. Thank you.